All right, so here we are, first session. My name is Dr. Calabrese. I will be teaching the anatomy and physiology. I'm going to start by kind of taking you through the like the basic setup of the course in Canvas. So if have you used Canvas before?
Yes.
Okay, all right. Well, I'm going to go through some of the basics just in case there will be people watching later who haven't. Um, So when you open up Canvas and enter into the class, it's going to look something like this. You see the most recent announcements here at the top of the page. So hopefully we'll be reading those as they come out. So I will be posting announcements that has important information like the passwords to quizzes and the links to these videos that we'll be recording. So all of that will be in the announcements, which you can get to just by clicking here, or you can go to the announcements tab on the left-hand menu here, and that'll take you to all the announcements. I'm going to go back to home and scroll down a little bit so that we can see the kind of access to all the basics of the course here. So here's a tab for your instructor. So if you click on that, it'll take you to my contact information, my office hours, all that information is in there. Um, in the assignments tab, that's where we're going to be able to, I'm going to click on it so we can see where you can access all the assignments. This is just one of several ways you can get to the assignments for the course. So if we start off on Unit 1 here, we see it gives us a little introduction to the topic, and then we can click on Unit 1 required assignments, and it will give us everything that's going to be due this week. Right, so we've got MyPath learning content, we've got a discussion post, there's a quiz and lab activities, right? And that's going to be the four basic things that are due every week. So there's always going to be learning content, discussion, quiz, and lab activities. So I'll show you how to access each of those. So you can obviously you can access them here by clicking on any of these links. If I scroll up and go back to home, there are other ways to get to those as well. One of the other ways is through syllabus. So if you click on syllabus here, and we scroll down, then we can also get to each of the units. If I click on Unit 1, again, there's access to all of those assignments. And here it tells you how many points they're worth. So the discussions are always worth 20 points. The learning content's always worth 25. Quizzes are worth 30, and your lab activities are worth 40 points each week. And if I click on, let's click on a couple other ones. If I go to Unit 3, we can see that in Unit 3, there's going to be an exam, right? So that's worth more points than the quiz. So that's 60 points. Exams cover more than one unit's worth of material. So that Unit 3 Exam 1 will cover Units 1 through 3. So it's a cumulative exam. Right. Back home to make sure we've kind of hit on everything on the home page here. There's also a link here to the library, and I highly recommend using the link to the library. You're going to be writing a lot of discussion posts for this course and probably all of your online courses. So the library is a good jumping off point to do your research. So you can access databases, you can access virtual textbooks, you can access a lot of journal articles, and also access to librarians, which is super useful. So if you have any problems with like finding the right sources, or even just simple things like knowing how to cite properly in APA format, then that information is here in the library. Citation help in APA style is there. And there's a direct Ask the Librarians link if you need help with something. Okay. All right. And so that's pretty much it here for this home page. If we scroll down, Again, there are some links here that are kind of useful, links to discussions and assignments, but there's multiple ways to get to each of these places. So I'm going to skip down to the syllabus tab here. All right, so that again takes us to the same place where it's breaking down what's due every week. And if I scroll past that a little bit, we can get to the grading scheme. And this course, like all courses here, is 1,000 points per term. And your job is to pass the course. So for this course, I believe you're going to need an 80% to pass the course. So we're going to try to keep at least 800 out of 1,000 points for the entirety of the term. And you can see those points are broken up in lots of different areas. So it's not like you just have one or two big tests. There's a lot of different ways to accumulate points. And it's my strong recommendation that you take advantage of all of those different avenues, because it's, again, getting 800 out of 1,000 can be challenging if you're, leaving something out. So try to make sure that you're doing your discussions, doing your lab activities, doing your learning content, and obviously taking the quizzes and exams. Try to get that 800 points. We'll talk about a makeup policy here, and you're, obviously free to read through this in more detail, but basically what it comes down to is if you're going to be late on an assignment, if you know that something's coming up in your life that's going to make it more difficult for you, or there's an emergency situation that's taking up a lot of your time, it's best to notify me as soon as possible. Right. And so, and again, my email address is in the faculty information tab here. But it's just acalabrese@herzing.edu. You can get a hold of me that way and let me know in advance if you're going to be late on something, because then if you've got documentation for your absence, like a doctor's note, military service, something like that, then I can grant you an extension of up to 1 week to get assignments in. There are no assignments can be turned in past 1 week overdue, even with a doctor's note, just because we get so much done in such a short amount of time, right? This is a very compact course. So if you're more than a week overdue, chances are you're not going to be able to catch up. So we try to keep you on task as much as possible. So that's the late policy. Let me go down here. The next link on the menu here on the left is modules. So if I click on modules, the first one here is read me first. So that's there for a reason. So there's a link to navigating the course, an introduction to Labster, which I think is really useful. So if you've never had Labster in any courses before, I would work my way through that introduction. Information about MyPath. MyPath is your learning content. I'll show you that in a minute. And then there's a link to a syllabus that I've updated for this course. Okay. All right, so that's the read me first. After you've done that, then we can get into the Unit 1 module. And if I click on that, do for you. And it's taking us to the same kind of list of assignments here. So we've got MyPath, we've got a discussion, a quiz, and a lab. I recommend starting every week with the MyPath. So I'm going to show you how to use this. So if I click on that and scroll down, it'll say load the Unit 1 in a new window. Got that. Okay. So when we're here, we can see on the right-hand side, we've got a learning map. So a list of 12 activities here that you're going to have to work through. If I hadn't already played around in this course, it would say begin, but here it says resume. So what we'll do is I'll just start at the beginning here and look at this learning map. So at the very beginning, overview of anatomy and physiology. And notice here's all 12 of those topics for this unit. So I'll click resume. And when we open this up, it's gonna, I want you to think of the MyPath as being your textbook, right? So this is your textbook. So it's really important to live here, right? You're gonna spend a lot of time reading this book. Anatomy and physiology is not easy. There's a lot of material to learn, a lot of scientific terminology. So we need to spend a lot of time with it to get the most out of this. But MyPath, I think, breaks it down into these nice kind of bite-sized chunks that makes it easier to process, at least in my opinion. So it'll start you with an introduction, and then you can go to the next little section here and go to the third one. And then as you're reading all of these, um, you'll continue to hit next and read through, one little paragraph at a time. There are going to be videos embedded in here. If you ever come across a video that's not there or is not functional, please let me know. Let me know the unit and the topic that you found it in. So in this case, unit 1, and the topic is overview of anatomy and physiology. So if you ever find something not working, let me know. We'll continue to click through, next, and see it's keeping track of my progress on the side. I'm getting green check marks for having read through that. All right, and then when I get to the end, at the end of each topic is going to be knowledge check questions. So these questions are going to be about the information you just read above in the topic, and you're going to want to do your best to answer these questions correctly in order to earn points. So this reading your textbook, you actually get points for. So this is worth 25 points every single week for reading through all these topics, or at least a maximum of 25 points every week if you get all the questions right. So you go through, you'll answer the questions. It's usually 5 at the end of each topic. And then when you're finished, you'll click exit to leave that topic. Right, so a group of similar cells, do that. Show next question. Basic levels of organization from smallest to largest. Do that one. All right, I'm not even going to read it. I'm just going to click one so we can see what happens if I get it wrong. So if I get it wrong, it'll tell me I got it wrong, and then you can view solution, and it'll tell you the correct answer. All right, click on the next one. And the last one. So I just want to show you what it looks like as we finish this. So I got 4 out of 5 right. I'll click next. It'll take me to a little summary of what I just read. Got the references. And then it says your lesson data has been recorded. So that's crucial, right? It's recording the fact that you've gone through this lesson, that you've answered 4 out of 5 questions correctly, so for 80%. And then we can click close. And then when I go back to the learning map, I can see that I got the light green here with a little check mark inside. So I've done pretty well, but I haven't fully maximized everything I can do in that. So I can always go back in and hit practice and do it again, right? So you can continue to explore the same content over and over again. And your goal is going to try to get mastery. So mastery will be the dark green with a little star in it that lets you know that you've gotten the best you can for that particular topic. And again, do them as many times as you want, right? There's unlimited access to this, and it'll continue to record your best score. There's a menu in my way and I can't get back. All right, so I'll go back to here and look at the modules again. All right, so that's the MyPath. Again, 25 points every week. That's always going to be due Sunday night by midnight Central Time. So make sure you get all that done. Next one here is the discussion. All right, so if we click on discussion, it takes us to this page where we can see instructions. on what you're supposed to be doing. You'll read through those instructions. And then your job is going to be to post an answer, that encapsulates all the questions here. And with cited references in this discussion board. So we can see Kierlyn has posted already with references. Right. I've posted in here basics on how to write a discussion post. So key factors on how to write it are going to be, have your title with your name and the unit. Introduce yourself, especially since this is the first one. It's good to get to know each other in the classroom. Make sure you're answering all of the questions asked in complete sentences, right? So try to avoid little phrases. Make sure we've got a complete sentence with appropriate capitalization and punctuation. Answer the questions in paragraph form. And do your own research, right? So make sure that you are researching the answers to these questions and that you've got solid references behind your answers, right? So that means looking up in reputable websites the answers to these questions, not just like, so-and-so's blog about anatomy, right? Make sure it's a really good, like a government source or a university source, something that lets us know that this is a accurate representation of the information. When you're doing that though, don't copy and paste. So this has to be written in your own words. We're going to try to avoid plagiarism at all costs because there are consequences for that. So write it in your own words and cite your sources. Right? So try to avoid any plagiarism there. Right. And then, include something that you found surprising or you didn't know about, or maybe this is your first time learning about something, or maybe it led you to further questions that you can pose for your fellow students. And then make sure you've got your references at the end. So we can see we've got some other posts going on here, which is great. Right, so that discussion post, this is going to be due every Wednesday by midnight. So you have an initial post due by Wednesday at midnight. So every week, make sure you're kind of getting on that at the beginning of the week. Everything else is due at the end of the week, Sunday night, but the discussion post, that initial one's due by Wednesday. So make sure you're working on that. And then also notice that you have to do a peer response. If I scroll down here, please post 2 or more peer responses. That means you're going in here, you're reading other people's posts, and you're responding to them. And this section gives you guidance on what a response should look like, right? So again, the responses are going to be, you should write in full sentences, you should write in your own words, and try to connect, what your peer said to the content for the week. Ask open-ended questions. So the idea is to post 2 peer responses, so 2 different peers every week. And the peer responses are worth 50% of your grade, right, for the discussion. So if discussions are worth 20 points every week, that means the peer responses are worth 10. So make sure you get those in. The peer responses will be due by Sunday night at midnight. So I don't expect you to have responses if everyone hasn't posted yet, but initial post by Wednesday at midnight, peer responses by Sunday at midnight. Back to modules. All right, so we got learning content, discussion. There is a quiz. So the quiz, 30 points, but 20 questions. These quizzes, again, they're going to be due Sunday night by midnight. You can hit load unit 1 quiz in a new window. It'll take you to a launch page that you have to have a password to access. So I will be posting the password to the quizzes every week on Thursday. So on Thursday, I'll post the password and you'll have from Thursday until Sunday to complete that quiz. All right. You've got a timer, right? So there's only 40 minutes available to you to take the quiz. So make sure that when you decide to sit down and start it, you're going to have time to finish it because you've only got 2 attempts. Okay, so you take it once and then review your answers, and then you have a second chance to take it. But if you blow through both of those attempts, then you don't get a third chance. So make sure you've got adequate time, you've got a good internet connection to sit down and take those quizzes when it's time. Right. Your best score will be recorded. So if you did better the first time, we'll take that score. If you did better the second time, we'll take that score. Let's see, what else? And then the lab activities. So if I click on lab activities, we can see that there are 4 different activities for this week. So you'll click on these. It'll take you down here where it'll say load this in a new window again. And hopefully this works. I don't know if because I'm in student view, if it'll let me do it. Okay, so that works. So it loads this. When you're ready, you'll hit play. And it'll take you to this window here where it's loading a simulation. This could take a while. So make sure when you're, again, doing the lab that you've got a good internet connection and you've got time to allow for the simulations to load. If you switch pages at this point, it will not continue to load. So you have to unfortunately keep it on this tab until it's loaded to 100%. And then you can enter in. So there is a voiceover that goes with this. So there's like a little robotic lab assistant that will guide you through. If you don't want to listen to her, then you can mute and just read the instructions as well. So that's an option. But you'll have to finish each simulation to earn all the points from it. And it'll guide you what to do as you're going through it. So you can see this one's still loading.
Are the labs like the reading content where you can go back and do them again, or are they only a one-time deal?
That's a really good question, and I'll double check, but I believe you have 2 attempts on the labs. So if you didn't like your first score, you have a chance to do it again. Yeah, it's a really good question. I don't know if I have the patience to wait for it right now. But once you get into it, it'll explain to you, oh, we got to start. All right, so if I hit start, all right, so you can see what you get, this little robot guy who's going to sort of take you through and explain to you what to do as you do it. And notice I exited out, so it takes over your full screen when you launch it. To get out of that, I just hit escape. And then you can go back here and click resume if you want to jump back into it. Okay, so that is the labs. And again, notice that there are 4 of those labs for this week. So make sure that you're giving yourself the amount of time to get all 4 of those lab assignments done by Sunday at midnight, right? So pretty much everything due by Sunday at midnight. Let's see. Over here on the left, again, we've got access to the Labster. So if I click here, it'll take you to a Labster dashboard, which will show you the scores that you've earned on, right? So it'll show me, hey, I started this lab, but I didn't make it very far. Current progress, 0%. So I don't have a score yet, but it will show you all the activities you've done attached to a score. Right, and then there's also a grades tab. So if I click on that, it'll show me, my grades on everything I've done so far. So the discussions, the labs, and how many points they're all worth. So you can see your kind of running total throughout the course. Again, goal is to make it to 800 points by the end of the term. One more thing I want to show you here in modules, and it's not showing up here yet, so I'm going to go through in these live sessions, I'll go through a PowerPoint every week. I will put those PowerPoints in the modules section. So if you ever want to go and review a PowerPoint, you'll be able to find it and download it there. Any questions about Canvas or MyPath or Labster? And I am going to bring up the PowerPoint. All right, so there is a lot of material this week. So if you started to poke around in the MyPath, you probably noticed that it's a ton of material. And this is an accelerated program, right? Which means we have to pack a lot into a really short amount of time. So there is going to be a lot of onus on you, the student, to make this happen, to get through all this material. So I will spend some time with you kind of going over this. So my PowerPoints are going to mirror the learning content. So to kind of help guide you into understanding what that learning content is about. I recommend, doing both, doing the learning content, obviously, because that's worth points, but then reviewing the PowerPoints or listening to my lectures so that you get kind of double exposure to that information. Okay. So this intro unit covers a lot of ground. So from the very basics of what anatomy and physiology are, we're going to cover all that, plus anatomical terminology and cellular anatomy and tissue types and the integumentary system. So it is a lot this week. But we'll start off with the basics. So what is anatomy and physiology? Anatomy is the study of structure. Physiology is the study of function. So, and we need both, and both kind of go together. So we need to understand the structures of the body and how they work. So, and structure mirrors function in a lot of ways. So we've got, like looking at the first example here, the skull. So the structure of the skull is that it's hard, it's bony. It serves a protective function. So it protects what's underneath it, which is your brain. Then we've got another structure here, these tiny little air sacs of the lungs. Those have a very different structure. They're very delicate, they're very thin, microscopic structures, and that's because they serve a very different function, right? They need to allow for gas exchange across these very thin membranes to get oxygen into your bloodstream and carbon dioxide out of your bloodstream. So we're going to be looking at human anatomy and physiology at all different levels. So we're going to start at a very microscopic level and look at sort of how atoms come together to form molecules, how molecules can become larger molecules, macromolecules, how those become the components of your cells, how cells come together to form tissues, tissues come together to form organs, organs to organ systems, and then finally the full organism. And we'll look at why it's important to understand what's going on all different levels of organization in order to really understand how the body works. So your cells are going to be the basic unit of life, right? So, and you've got trillions and trillions of cells in your body, and we can break them into categories of different cell types. And you've got about 300 different types of cells, even though you started off as one, right? You started off as a zygote, which was just one single cell. One became two, became four, became eight, became 10 trillion over time. And the process by which they became these kind of unique cells is differentiation. So differentiation takes you from being sort of like a generic cell type stem cell to becoming something very specialized, right? Like we can see here, we've got cells here that look like they're on the surface of the skin, right? So those cells have a very different job than the cells down here, which look like they're in the lining of the intestines, right? Those are very different cell types that have very different jobs. And they became that particular type through the process of differentiation. So for our levels of organization, we've got all those different types of cells. We can put them into categories of 4 different tissues. So a tissue is just a group of cells that have a common origin that work together to perform a common function. And so we've got 4 major categories of tissues: epithelial tissue, connective tissue, muscle, and nervous tissue, right? And there are lots of different cell types that fall into each one of those 4 categories. Your tissues are going to come together to form organs. So every organ in your body is composed of 2 or more different tissues. And in fact, most of your organs are composed of all 4 tissue types. So for example, here is a cross section of your skin. So your skin is the largest organ in your body, and it's composed of epithelial tissue, connective tissue, muscle tissue, and nervous tissue. So it's got all 4 tissue types represented there. Right? Same is true for most of your organs. Right? And then an organ system is just a group of organs that have a common function. So they're working together for a particular goal. The example here is the digestive system. So digestive system's job is going to be to ingest food, to break it down, digest it, absorb the nutrients, and then eliminate the wastes, right? So in order to do all of those jobs, we need different organs that are all kind of working together to accomplish those goals. So there are a lot of different organs in the digestive system to get that done. So major functions of life here, and these are, kind of generic for a lot of life, but specifically for humans as well. So there has to be exchange with the environment. So that means we bring things in from the environment, and that's usually nutrients, and then we eliminate things back to the environment, and that's going to be waste. So the systems that help you accomplish that are digestive, respiratory, and urinary. We need fluid transport within the body to help us control where certain concentrations of liquids are moving and moving electrolytes around. So cardiovascular, lymphatic, and urinary system help with that. We also need structure, support, protection, and movement. That's mainly coming from your skin, your muscles, and your bones. And then control and regulation. So this is, the control center. What's encouraging the body to maintain itself, and that's going to be the nervous system and the endocrine system are both have a control function. Right, and the idea is all of these systems are going to work together to maintain homeostasis. So homeostasis is just the maintenance of like stable internal conditions, right? It's the kind of happy place where your body wants to be. And there are a lot of different ways we can measure homeostasis. So a really common one is just to think about body temperature, right? We're warm-blooded animals. We maintain, a body temperature around 98.6 degrees Fahrenheit, right? So that's what our body wants to maintain. And if we go too hot or too cold, we've left homeostasis, and the body will try really hard to get back to homeostasis, right? So, but that's not just temperature that does that, it's pretty much everything. So you have to maintain the correct pH of your blood. You have to maintain the correct calcium levels and potassium levels and sodium levels. And all of those things are maintained within a very narrow window. So failure to maintain homeostasis leads to sickness and eventually death. So death, the only cause of death is failure to maintain homeostasis. There's lots of ways you can do that, but the failure to maintain for an extended period of time will eventually lead to death. And there are constant challenges to maintaining your homeostasis. So there's external challenges, internal challenges, stressors from the environment that are going to, something as simple as walking outside on a hot day. That's a challenge to your homeostasis, right? Your body has to figure out a way to cool itself down so that you don't lose consciousness, essentially. So there are a lot of mechanisms to help you to do that. Right, and the main mechanism by which we maintain homeostasis is called a feedback loop. Right? So a feedback loop has three components to it: a sensor, a control center, and an effector. A sensor helps you detect any change from homeostasis. Right? So for the example I just used of walking outside on a hot day, you've got sensors that are able to pick up changes in temperature and let your body know that you're too hot, right? So the sensor picks up that information, it senses it, and sends it to a control center, right? In the case of humans, the control center is going to be the brain for the most part. So the brain will receive that information and decide what to do with it. So if it receives information that the body's too hot, then it's going to send a response to an effector. So the effector is anything that's able to respond to that information coming from the brain. So in the case of a hot day, the effectors might be your sweat glands, right? The sweat glands will receive a message from the brain that it's time to start sweating. As you produce sweat, it evaporates and it creates an evaporative cooling that cools your body temperature down, right? So sensor, control center, effector, always in that order, right? And eventually then hopefully we get back to homeostasis, at which point the sensor can shut off and it's no longer sending that kind of warning signal to the control center anymore. Yeah, so, and the sensors could be lots of different things, right? We've got thermoreceptors that are picking up on temperature. You've got chemoreceptors that can pick up on, your carbon dioxide levels or your oxygen levels or your pH. You've got baroreceptors that pick up on your blood pressure and send those warning signals to your brain that, hey, we might be inching away from homeostasis. We got to get back to it. Okay. Again, the control center receives the information from the sensor and creates a command that goes to an effector. Right? And the effector could be a lot of things. It could be something as simple as a skeletal muscle, right? If the sensation is pain, like, hey, I stepped on a tack, then that sensation of pain goes to the brain sends a message to an effector, skeletal muscle to pull away from the pain, right? To get away from the thing that's causing that reaction, right? So lots of different types of effectors. All right. So what I just described, where we are trying to reverse a change, so stay in our kind of happy place, that's a negative feedback loop. So negative feedback says, okay, if we're creeping away from homeostasis, I got to get back to it, right? So that's a negative feedback loop. And that's mostly what your body does. A positive feedback loop is when you accelerate a change away from homeostasis. All right, so that acceleration away, not super useful. Your body does it in a few instances. Blood clotting is a positive feedback loop, right? And it's one of those that we only want blood clotting to happen in a very narrow set of circumstances, right? Because if blood clotting happens when you don't want it to happen, that's a big problem. And childbirth is the example on the slide here. So that would be a positive feedback loop that keeps accelerating away from homeostasis until a child is born, and then it goes back to normal. So most homeostasis is maintained with negative feedback. All right. So now we're kind of switching gears to get into anatomical terminology a little bit. So we are going to have to learn a lot of terms throughout this course in order to understand how the body works. And we'll start by just understanding standard anatomical position. So standard anatomical position is how this woman is standing here. So she is standing upright, feet on the floor, facing forward, palms or arms at her sides, palms facing forward, head level, eyes facing forward. So that's going to be our standard anatomical position. And that's what we use as sort of a reference point. So that when we say, the head is superior to the torso, we know that the head is above the torso in standard anatomical position. That doesn't mean in every position the head is above the torso, but we can just refer to it as being a superior structure because it's above the torso in this position. All right. And then we've got a lot of regional terminology. So I'm not going to go through every single one of these terms, but this is a good place to make flashcards. So if you're not familiar with any of these anatomical terms, now's the time to learn them. So if I'm referring to the carpal, you should know I'm referring to the wrist. Here's examples of some of the important ones. So the skull is cranial, the neck is cervical, elbow is cubital, wrist is carpal, patellar refers to the front of the knee, orbital, the eye, thoracic for the chest, and inguinal for the groin. And you can see there's a lot more sort of listed over here on this image. All right, here's another handful of them. Right, and then there's the ones on the back of the body as well. So it is important to put these to memory because it's going to help going forward just to have these in your arsenal, right? And know what these terms mean so that you don't have to stop and look it up every time one of these terms comes across your screen. All right. So standard anatomical position is our reference point, but clearly there are other body positions, right? So we refer to someone laying face down as being prone, someone laying down face up as being supine. Right? And then considering standard anatomical position, we can use directional terms to describe where things are in the body. So superior is anything above or towards the top. Inferior is anything below or towards the bottom. And these are relative. They're relative terms. So if I'm looking at this body here, I can point out the stomach. So here's the stomach. The stomach is inferior to the heart, right? It's below the heart, so it's inferior. But the stomach is superior to the colon, right? Because it's sitting on top of it, right? So these are always relative terms. All right, and then we've got anterior and posterior. So anterior towards the front, posterior towards the back, again, relative terms. Medial and lateral. So medial is towards the midline here. So the heart is medial to the lung. The lung is lateral to the heart. And then proximal and distal. So proximal and distal usually refer to your limbs or your appendages. And proximal means closer to the point of origin. So like in the case of your arm, the point of origin is the shoulder. So the shoulder is proximal to the elbow. The elbow is distal to the shoulder, right? But the elbow is proximal to the wrist. The wrist is distal to the elbow. So always relative to one another. Okay. And then we can, again, usually referring to the limbs, we can refer to ipsilateral and contralateral. So ipsilateral is two structures that are on the same side of the body. Contralateral would be opposite sides. Contra meaning against, lateral meaning side. All right, and then superficial and deep. Superficial, closer to the surface. Deep is deeper within the core of the body. Again, relative terms. So obviously the vessels in here are deeper than the lung itself, but we can also look at this muscular wall and say that's superficial to the lung. All right. And similar to superficial and deep, we've got visceral and parietal. So visceral and parietal usually refer to membranes that cover some of your vital organs. So for example, we've got a membrane called the pleura that covers the lungs. The pleura has two layers to it, a visceral layer that actually touches the organ, and then a parietal layer, which is more superficial. It lies against the body cavity. Right? So, and visceral is always going to be deeper. Parietal is always going to be more superficial. All right. We can also look at body planes. So a lot of the times when we're looking at structures within the body, we're not seeing the body as a whole. You're seeing an image of a part of the body. And in that case, it really helps to know what body planes you're looking at. So a sagittal plane is one that cuts through the body, separating left from the right side. So in this case, the sagittal plane would be right here, right? Separating left from right. And we could have a mid-sagittal plane like this one that goes right through the midline, or parasagittal, which still separates left from right, but not in equal halves. Okay. We could also have a transverse plane. Transverse plane is just any plane that cuts the body, separating top from bottom. Again, it could be through the umbilicus here, or it could be, separating the top of the head from the bottom of the head. So it could be anywhere. And then frontal or coronal section separates the front of the body from the back of the body. So if I turn my head sideways, it would be a line cutting like this, so separating front from back. Okay, so there's more pictures of those. And here's an example of it on just a body part instead of looking at the whole body. So in a sagittal section here, separating the brain in two halves, left from right. Frontal separates front from back. And then a horizontal or transverse separates top from bottom. All right. I told you we were covering a lot today, so we're getting into what's an acid and what's a base and what is a measurement of pH. So it is important to know. So pH is a scale that measures basically the concentration of hydrogen ions in a solution. Right. So the more hydrogen ions in a solution, the more acidic something is. So really, something that's very acidic is acidic because it has a lot of hydrogen in it. The pH scale actually measures the inverse log of the concentration of hydrogen ions. So that means, that sounds confusing, but all that means is the lower the number, the more acidic a solution is. So we start at neutral, which is 7. And 7 is 7.000 is totally neutral. Anything below 7 is acidic. And the further we go below 7, every single one of these numbers, it becomes 10 times more acidic. So something that's a pH of 6 is 10 times more acidic than a pH of 7. If we go down to 5, that's going to be 100 times more acidic than a pH of 7. So it gets increasingly more acidic at a logarithmic or exponential scale. Right, and the opposite happens when you go the other way. So if you're moving from 7 to 8, something that's a pH of 8 is going to be 10 times more alkaline or basic than something that's a 7. Right? And notice right here in the middle at 7, we've got an equal concentration of hydrogen ions and this other guy, this is a hydroxide ion. Right? And if we put one hydrogen and one hydroxide together, we get water, right? Which is why water is our neutral thing, which is right in the middle, right? If we've got more hydroxide ions, then we become more basic. So just a note here of different kind of body solutions and where they lie on the pH scale. So 7 is neutral, so that would be pure water. At a pH of 7, but look at the acidic side here. So pH is way down here at about a pH of 2 or 3. We find your stomach contents. So your stomach is full of hydrochloric acid. It needs to be at a pH much lower than the rest of the body in order to do its job. So just because a pH of 7 is neutral doesn't necessarily mean it's good for somewhere in the body, right? If your stomach were a pH of 7, you would be very sick, right? It would not be good for you. If we creep up here a little bit closer to neutral, we get to urine. So urine is usually a little bit more acidic because your urine is one of the places that we get rid of extra acids. Right? So it's a waste product that allows us to get rid of those acids. Up here we see blood. So blood is just a little above the neutral point. So it's actually alkaline, and your blood should be ever so slightly alkaline. So between 7.35 and 7.45, right? And that is very tightly controlled to maintain that homeostasis. So anything below 7.35 puts you in acidosis. And acidosis causes a lot of problems. You're very sick. Alkalosis is if your blood pH is above 7.45. So again, very tightly controlled. Okay. And we can manage that tight control because we've got buffer systems. So buffers are things that are they're chemicals that are going to be able to maintain a pH without having large swings from acid to base. It kind of helps to hide extra hydrogen ions or extra hydroxide ions so that you can maintain that sort of stable pH. Acids and bases neutralize each other. So if you add a really strong acid to a really strong base, you end up with something neutral in the middle, like water. All right, so I know we covered a lot so far. I'm gonna pause for a second and see if there are any questions.
This can be downloaded, correct?
Yes, so this is all being recorded. I'll post a link so you can watch it later, and I will post this PowerPoint as well in the modules so you can download all the slides. I know it's a lot. So we'll move on to cellular structure and just kind of look at the basics of a cell here. So your generalized cell has a plasma membrane. It's got cytoplasm with organelles, and it's got a nucleus. So a nucleus is the largest organelle, usually in the center of the cell. So the cell membrane or plasma membrane, that's the boundary that separates the inside contents of the cell from the outside, and it is an incredibly important structure. So it is a semi-permeable membrane, which means that certain things can cross that membrane, but other things can't. And that's what helps give the cell its integrity, right? It helps it stay alive, basically, because it can determine what it wants to come in and what doesn't. So structure of a cell membrane is going to be the same for all cell membranes. They all have a phospholipid bilayer. So a phospholipid is one of these molecules here that looks like a little ball on top and two little legs coming off of the bottom. And it's a bilayer because there's two layers of those phospholipids with their little tails pointing toward each other. And then the phosphate group heads, the ball part, being on the outside of the cell or on the inside of the cell. The little legs here are the little tails. Those are fatty acids. Right? So a phospholipid is a molecule that's partially fat, which is really important because that's what creates the protection of the membrane. So it's a little fatty bubble, basically, that surrounds all of your cells and helps protect them. Right? Embedded within these phospholipids, we've got proteins. So here's a protein that crosses all the way through and acts like a little tunnel or doorway. We've got other proteins that just kind of stick out one side or the other. And they all have different jobs in allowing the cell membrane to perform its particular functions. Skip by those. And we'll talk about transport because I want to get through as much of this as possible, and I'm realizing we're not going to make it. So transport processes, We can divide into being either passive or active. Passive transport, and by transport, I mean moving across the membrane. So moving either something into the cell or something out of the cell across that phospholipid bilayer. Passive processes don't require any energy. Active processes do, meaning we would have to pay some energy to move something across the membrane. Right. So the most simple of the passive processes is diffusion. So in diffusion, the substances just want to move from a high concentration to a low concentration. So if we've got a higher concentration of something on one side of a membrane and a lower concentration on the other, those particles are going to want to roll down their concentration gradient until they're even on both sides, right? That's all diffusion is, right? Just high concentration to low concentration until the concentrations even out. Right? And simple diffusion happens when things can just slip right through that membrane, so cross from one side to the other. Facilitated diffusion is also passive, but in this case, it uses a doorway. So we're using a protein like passageway for things to move across the membrane. They're still going from high to low concentration, still no energy involved, except there's a doorway that speeds up the process. So there's simple diffusion. High concentration of whatever this is here, low concentration here. Those molecules, if they can fit through, will eventually even out on either side of the membrane. In facilitated diffusion, again, they will still even out, but they're going to be using some sort of protein to get from one side to the other. So here's an example of that. So going through a channel or going through a carrier protein to go from high concentration to low concentration. So now we get to a really important type of passive transport, which is osmosis. So osmosis is, it's still a passive process, but it's a very specific kind of diffusion. So this is the diffusion of water only. So the diffusion of water across a semi-permeable membrane from an area where we've got a lower concentration solution to a higher concentration, which sounds backwards. But what the water is doing is moving across the membrane in order to even out the concentrations. So it's going to help water down the higher concentration until we get even concentrations. Right. So this is happening in your cells all the time. Right? So osmosis is always happening. Can't stop it from happening. It's just water moving across the membrane. And we refer to the concentrations of the different fluids like within your cell and outside of your cell as tonicity. So tonicity refers to basically the concentration of salt inside versus outside the cell. Right. Ideally, we want your cells to be living in an isotonic solution. So isotonic meaning we've got the same concentration of salt inside as we do outside. So in this situation over here, we've got about the same amount of salt inside as outside. And what's going to happen if we've got the same concentration of salt in and out is that water will move across that membrane in equal amounts. Right? We can see that there's water going out and there's water going in. Right? But the water going out and the water going in even out. So everything stays even. We're isotonic and the cells are happy. They're in their normal state. This is a hypotonic solution. So hypo meaning less. So what's happening here is the solution that the cell is in has less salt than the inside of the cell. Right, and that's kind of bad news for the cell. So what's happening here is since we've got more salt inside and less salt outside, the water wants to chase that salt. Water always chases salt. So wherever you've got a high concentration of salt, water is going to follow. Right? It's the same reason as why you feel like bloated if you've eaten a really salty meal, right? The water wants to kind of dilute that salt so it rushes towards it. So this is not a great situation for your cells to be in because we've got more water rushing into the cell than out. So that can cause the cell to swell. And eventually, if it swells too much, it could burst, right? So that the bursting of a red blood cell is called hemolysis, right? Hemo meaning blood, lysis meaning to break apart. All right. The opposite of that is a hypertonic solution. So hyper meaning more. So in this case, we've got more salt in the solution than we do inside the cell, right? And so the water will always chase salt. So in this case, water leaves the cell to try to dilute the salt that's on the outside. And this is very bad for the cell because if the cell loses all of its water, it's going to shrivel up and die, right? And so that process is called crenation. So red blood cells in a hypertonic solution will experience crenation, right? So this is why your kidneys are so important because they help to maintain just the right amount of salt and the right concentration of your blood. So getting rid of enough water or getting rid of enough salt so that your cells are in an isotonic solution so that they don't swell or crenate. Keep them happy. Right? So, but all that's passive, right? So the osmosis of that water moving across those membranes to even out concentration gradients, that's a passive process. Another passive process is filtration. So filtration, instead of evening out concentration gradients, filtration evens out pressure gradients. So this is movement of some water from a high pressure situation to low pressure, right? And that's just the way that water wants to go. It always wants to go from high to low pressure. So this is what your kidneys use to be able to filter the blood. They use filtration. All right. So all of that was passive. We'll move on to active. So active transport is when we have to use energy to move something across the membrane, usually because we're going against the concentration gradient. So if we've got, say, too much salt on one side of the membrane, we can use active transport to get even more salt on that side of the membrane. So it'll actually create a gradient instead of eliminating one, right? Passive eliminates a gradient, active creates one by creating different concentrations. We'll explain why that's necessary as we get into talking about how your muscles and your neurons work. But the main mechanism we use to do this is a structure called the sodium-potassium pump. Right? It's just a protein that sits in your cell membranes that is going to move sodium ions out of the cell and potassium ions into the cell. So you can see we're moving 3 sodium ions out for every 2 potassium ions that come in. And every time we do that, it costs us 1 molecule of ATP, which is like your cell's money. It's currency. So that because we have to use energy, that's what makes it active transport. All right. And then the last type of transport here is bulk transport. So bulk transport is when you're moving something across a membrane that is too big to fit between the phospholipids, too big to use a carrier or channel protein. So we actually have to form a vesicle. So vesicle is like a little like fat bubble that butted off the membrane. So remember I said your cell membranes have a lot of like those fatty acid tails. We can form a vesicle from that membrane, just like a little bubble. And we can use that to merge with the membrane and either bring stuff into the cell or push stuff out of the cell. If we're bringing stuff in, that's endocytosis. If we're pushing stuff out, it's exocytosis. So endocytosis brings things in, and there are 2 different categories here, phagocytosis and pinocytosis. So phagocytosis would be bringing in something large and solid, like when your white blood cells eat a bacterium, they do that through phagocytosis. pinocytosis is usually for like smaller liquid intake versus solid. Right, and here's what that looks like. So basically, if we want to eat a bacterium, we have to wrap the membrane around it to form that little vesicle. And that vesicle used to be part of the membrane, but now it's a separate vesicle that has ingested this bacterium. All right, so that's the membrane. If we get deeper into the cell here, beneath the membrane, we have the cytoplasm. So cytoplasm consists of the cytosol, which is the fluid, and all the organelles that live inside the cell, which all have specific, an organelle just means a little organ. So they all have specific jobs inside the cell. So hopefully some of this is review from like maybe an earlier science class or biology or something in high school. But we'll talk about the different organelles briefly here. So endoplasmic reticula, you've got a few of these in each of your cells. So it consists of rough ER and smooth ER. Rough ER is called rough because it's got ribosomes on it. So these little like dots on the rough ER are ribosomes. It makes it look rough under the microscope. So because it's got ribosomes, whenever you think about rough ER, I want you to think proteins. This is where proteins are made. Smooth ER is attached to the rough ER, but it does not have ribosomes, so it looks smooth. Smooth ER is usually associated with fats. So this is where we synthesize and store fatty acids. All right, so those little itty bitty dots that were on the rough ER, those are the ribosomes. A ribosome is a site, oopsie, do that. That's a site of protein synthesis. So this is where proteins are actually assembled. And proteins are just made of chains of amino acids. So your ribosomes kind of help stick those together to form large proteins. All right. Then we've got the Golgi complex. So Golgi is this kind of like he looks almost like a stack of pancakes, these flattened sacks that sit on top of each other. The Golgi complex's job is to basically finish off the protein. So after a protein's been manufactured by a ribosome in the rough ER, it will then use a transport vesicle as like a cab that takes it over to the Golgi, and the Golgi will do the final touches. It'll kind of finish up the folding of the protein, add whatever final touches it needs, and then it'll send it out in another vesicle to where it's going. So you can think of Golgi as like the FedEx of your protein system. All right, and then lysosomes. So lysosomes, these are tiny little organelles that basically you can think of as like a garbage disposal inside your cells. They help with autophagy. So autophagy means self-eating. So when you have to break stuff down on the inside of your cells, it's going to be the job of your lysosomes. Okay. And then mitochondria. So these are super important organelles. Mitochondria are interesting because they're their own organisms, really, that kind of live inside your cells. They have their own DNA that's separate from your human DNA, and they used to be independent bacterial cells, but now they live inside your cells, and their main job is going to be to generate ATP. So they will use the food that you eat and the air that you breathe in to do a really good job of making a lot of ATP energy from that food, right? Without mitochondria, we would not be able to efficiently make use of the food that we eat. So really, really important, right? Peroxisomes, these are kind of like a lysosome, except they're more specific in what they break down. So they're good at detoxifying radical oxygen species, so free radicals, breaking those down so that they don't cause damage. All right, then centrioles. So centrioles are the origination point for your cytoskeleton. So your cytoskeleton is like the structure that maintains the cell, kind of holds it all together. And the main component of that is microtubules, which originate from the centrioles, right? They're also really important for cell division, things like that. So if we look at the cytoskeleton, we've got three major components. The biggest one is the microtubules. So microtubules are made of a little protein called tubulin, and they kind of create these like hollow beams that really support the cell. They're kind of like the scaffolding, like the steel beams that support a building, right, and kind of hold the thing together so it doesn't collapse. We also have microfilaments. So microfilaments are made of a protein called actin, and this is usually associated with the cell membrane and helps the cell membrane change shape. So when the cell, like for example, if we want the cell to pinch into two and divide, the cell membranes had to change shape in order to do that. They do that with microfilaments. And then the last one, intermediate filaments. These are usually made of keratin, but it depends on the type of cell we're in. These radiate from the nucleus to the cell membrane. And as they're going from the nucleus to the membrane, they basically hold all the other organelles in place. So keep everything from sort of like bumping into each other inside there. All right. Also, your cilia and flagella are made of microtubules, right? So, and not every cell has these. In fact, most cells don't. So cilia are short little hair-like projections that you have multiple of coming off the side of a cell. Cells that have cilia in the human body are going to be ones inside your respiratory tract. And the job of the cilia there is to help, they kind of serve as like a brush or oars that are pushing fluid. They're going to push mucus up and out of your respiratory tract. You also have them in the ventricles of your brain to help move cerebrospinal fluid. You have them in your fallopian tubes to help move an egg from the ovary to the uterus. So those are places where you have cilia. Flagella, there's only one human cell that has a flagellum, and that would be a sperm cell. All right. So let's look at how cells are held together, right? So your body is, like we said, is made up of trillions of cells. They have to stick together somehow. And there are different mechanisms that cells use to do this. But if we're looking at, say, epithelial cells, like the ones in your skin or your mucous membranes, they're going to be held together by tight junctions. So a tight junction is something like this, where we've got one cell membrane here and another cell membrane over here. And then we've got these proteins that kind of like zip them together. So think of tight junctions as being like a zipper, right? It's a really close association between those two cells, so nothing else can get through. Right? But we also have other types of junctions. So these anchoring junctions, like this desmosome. I think of a desmosome as more like a snap or a button, right? It's still holding two edges together, but there's gaps on either side, right? And then this last one, a gap junction, this is more like a tunnel that allows a connection between the cells. So products can go from one cell to the next cell through these little gap junctions. All right, we made it to chemistry. Any questions about what we've covered so far? Take some water. All right, so we'll jump into chemistry. Again, we're not going to make it all the way through this, but let's see if we can at least get through some of it. So this is really starting at the basics. So I mentioned that we kind of need to understand, in order to really understand how the body works, we need to understand it on a really basic level. how atoms come together to form molecules. So to start, we'll just look at an atom. So an atom is the most basic unit of matter for any particular element. Atoms have a nucleus. Nucleus is composed of protons and neutrons. And then they have a shell that surrounds that nucleus, and those are electrons in the shell, right? If an atom is neutral, then it has the same number of protons as it does electrons. Protons have a positive charge, electrons have a negative charge. So if we have the same amount of both, then they even out and we have a neutral atom, right? Each atom of a particular element is unique, right? And we can tell what element we're looking at by the number of protons in the nucleus, right? And the number of protons is going to be the atomic number. So if we look at the periodic table here, like our first one here, hydrogen, hydrogen has 1 proton. So any atom with 1 proton is hydrogen. That's the only thing it can be, right? If we stick with our organic stuff here and we look at carbon, carbon always has 6 protons. So it will never change. If we change the number of protons, we change the element, right? If we go up from 6 to 8, then we've just changed from carbon to oxygen, right? So the number of protons is the atomic number. Oops, neutrons are the other component of the nucleus. So neutrons help kind of make up the mass of the nucleus of the atom. The number of neutrons is variable, right? So if we look at a standard carbon atom, it's going to have 6 protons and 6 neutrons, so 6 of each. That gives us carbon 12. But it's also possible to have a carbon that's got 6 protons and 8 neutrons, right? That gives us carbon 14, which is an isotope. So it's the same element, but a different number of neutrons. That's what an isotope is. Same element, just a different number of neutrons. So it's basically like a heavier version of regular carbon. So atomic number, mass number. So the atomic number we said is the number of protons. Mass number is the sum of the protons and neutrons. So carbon 12 or carbon 14. The 12 or the 14 would be the mass number. The atomic mass is a little bit different. The atomic mass is always going to be a decimal, right? It's always going to be a fraction because it's sort of an average of the weights of all of the atoms of carbon in the entire universe, right? So it's looking at all naturally occurring carbon and saying most carbon is carbon 12, but there's other heavier carbons out there. So I think for carbon, it's like 12.066. So it's not, the mass is a little bit heavier than your average, or it is the average. All right, so that's the nucleus. And then surrounding the nucleus, we've got electrons. So electrons, we said, are negatively charged. They kind of orbit around the nucleus of an atom, just like the moon orbits around the Earth. Except we have different numbers of these electrons based on the number of protons that are in the nucleus. And they tend to form into rings, what we call shells. So the first ring around the nucleus of the atom usually holds 2 electrons, then the next one holds 8. And so whatever the outermost shell is, we call our valence shell. And usually that valence shell wants to be full, right? It wants to have the maximum number of electrons. So let's look at the carbon here, just for example, right? We got 6 protons, 6 neutrons. Our first shell holds 2 electrons. And then our second shell can hold up to 8, but in the case of a neutral carbon, it's only got 4 because we need 6 of these negative electrons to balance out the 6 positive protons, right? So those even each other out to give us a neutral carbon, right? There's still empty spaces there, so that carbon, he's going to want to fill those empty spaces, and he'll be able to do that by bonding to other elements. right? So a molecule is what happens when two or more atoms come together and form a bond, right? So they're kind of hanging out really close to one another, right? So this is a molecule of oxygen. So a molecule of oxygen has two oxygen atoms, and you can see they're intertwined here, and that's because they're sharing electrons, right? Those electrons are being shared between the two. And an ion. So an ion is just a non-neutral atom. So an atom that's no longer neutral, it has a charge. So it's either got a positive charge or a negative charge. And sodium is a really common ion. We can see it's got 2 electrons in its first shell, got 2, 4, 6, 8 electrons in the second shell. And then it's just got this one lonely electron in the third shell. So he's got 11 protons and 11 electrons. So he's neutral here. The problem is he doesn't want to have this valence shell with just one electron in it. So he will happily donate that to anyone walking by. When he donates that electron, now we're down to just 10 electrons for 11 protons. So we've got 11 positive charges and 10 negative charges. So they're not quite even, which gives us an overall positive. So this is a positive ion because he's gotten rid of that extra electron. Now he's happy because his valence shell is full, but he's got a charge. So he's an ion now. So I want to look at the ionic bonds while we're talking about that. So a positive ion like that sodium ion we just looked at is a cation. So cation is positively charged. An anion is negatively charged. So that would be if we had to get rid of, or I'm sorry, if we gained more electrons, then we would have a negative charge. All right, so, and that happens in this example for chlorine. So if we look at chlorine, he's got 2, 4, 6, 7 electrons in his valence shell, but he can hold up to 8, right? So he wants to gain an extra electron just to fill that shell and help keep him stable. So he will happily accept an electron that's been donated from somebody else. But when he does that, that makes him a little negative because now he's got an extra negative charge. So we've got a cation here and an anion here. So those two opposite charges are going to be attracted to one another, right? Opposites attract. So when we've got this situation where we've got a sodium and a chlorine really close to one another, they're going to want to stay close to each other. So they're attracted by those opposite charges. And that's what makes sodium chloride. That's what makes a salt molecule. They're held together by an ionic bond. So ionic bond is just opposites attract. Right. The other type of bond, which is much stronger, is a covalent bond. So covalent bond is instead of just donating and receiving electrons and then sticking near each other because of opposite charges, a covalent bond is sharing electrons. So you're literally using the same pair of electrons between the two different atoms that are bonded, right? So, and we can have different types of covalent bonds here. We've got a triple covalent bond, and that's holding these two nitrogens together. So this is a very strong bond. It's hard to break apart those nitrogens. Over here, we've got single covalent bonds between carbon and hydrogen, 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 hydrogen. Gives us a methane molecule, right? But these lines here indicate that this is a covalent bond. These guys are sharing a pair of electrons. So each line represents one pair of electrons. All right, and the last type of bond that we'll talk about is a hydrogen bond. Normally when we're talking about bonds, we're talking about something that's creating a molecule. But in this case, hydrogen bonds are attractions between molecules. So if we look at a water molecule here, a water molecule here, here, these are all water molecules that are slightly attracted to one another. So I won't get into the details of it here, but if we look at this, um, each water molecule consists of an oxygen and two hydrogens. So this molecule is covalently bonded together. But because the oxygen is so much bigger than the hydrogens, he's kind of like an electron hog, and he's holding the electrons closer to him than to the hydrogens, which makes the oxygen side of the molecule a little bit negative and the hydrogen side of the molecule a little bit positive. So what happens is the little bit positive side of this water molecule is attracted to the little bit negative side of this one, right? And they form this sort of like weak attraction between each other. And that's what a hydrogen bond is. It's just an attraction between adjacent water molecules. And that hydrogen bonding that happens between water molecules makes water really, really special, right? So water is our universal solvent. It's what we use to move things around inside our body. Your blood is mostly water, your lymph is mostly water, cerebrospinal fluid is mostly water, interstitial fluid is water, right? You've got all of this water in your body because it's really good at moving things around. Um, and one of the reasons why is because of that hydrogen bonding that it has. So there you can see the shared electrons. So there's a shared pair there, shared pair there, and both of those shared pairs are going to be slightly pulled towards the oxygen side, which makes the oxygen side a little bit more negative. Right? Hydrogen bonding is what creates surface tension on water. So it's what allows this little water bug here to kind of skim across the surface of water because water likes itself so much, right? It's really attracted to itself. It's also what, like, if you pour a glass of water and you overfill it to where it's got that kind of little bubble over the top and it's not quite spilling over the sides, it's able to do that because of the hydrogen bonding. It's because the water molecules love each other so much. You wouldn't be able to do that with another liquid, right? If you tried to do that with an alcohol, it wouldn't work. There's not enough water in that to adhere to each other. So we mentioned that. All right, so water, obviously really, really important, but we should mention some other important molecules in your body. So these are organic compounds. So an organic compound is one that's got carbon, hydrogen, and oxygen. Right? And there are 4 major types of organic molecules that we'll be covering. So those are going to be your carbs, your lipids, your proteins, and your nucleic acids. So you might notice those as being like your macros, right? Your macros that you eat in your diet. Right? That's also what you're made of, right? No surprise there. So carbohydrates are the first ones that we'll look at. Carbohydrates are always made of carbon, hydrogen, and oxygen, period. There's nothing else. They're just carbon, hydrogen, oxygen. They can be really small, individual little monosaccharides, like a simple sugar. They can be disaccharides, which are double sugars, or polysaccharides, which are really bigger carbohydrates, right? Carbs are important for, oops, I guess I don't have another slide. Carbs are important for energy, right? This is your main source of energy. A carbohydrate like a glucose molecule, a simple sugar, can be used by every single cell in your body to make energy, to make ATP. So carbohydrates are the simplest form of energy for your body. You don't have a lot of carbohydrate storage. So when you use carbohydrates, you don't have a lot of like built up carbs around your body. So this is why you need to eat them, right, pretty frequently in order to get carb energy. Okay. Lipids are another major organic macromolecule. They also contain carbon, hydrogen, and oxygen, except in different amounts. So look at these couple fatty acids down here. Notice that it's mostly a hydrocarbon chain. And then there's a couple little oxygens in there, but it's mostly carbon and hydrogen. So these fatty acids here are what makes fats hydrophobic, right? They're afraid of water. This is why oil and water don't mix, is because of this structure here. Right. There are different types of lipids that are important in your body. Lipoproteins are a combination of lipid and protein that's used to travel around in your blood, so that we can get lipids from one place to another. Because again, blood is mostly water, fat and water don't mix. So we kind of need to almost like cover those lipids in a little bit of protein to be able to transport them. So HDL, or high density lipoproteins, these are very compact or dense lipoproteins that are moving towards your liver in order to be converted into bile and passed out with the feces. It's usually referred to as your good cholesterol. LDL, or low density lipoproteins, they're bigger, they're not as densely packed, and they are moving towards your cells. Sometimes if you have too many of these, then this can lead to cardiovascular problems because they can clog up tiny arteries. All right. But your main lipid molecule that you're using for energy is a triglyceride. Right? So a triglyceride is this guy. He's tri because he's got 3 fatty acid tails and 1 glycerol molecule here in blue. Right? So this is your energy source from fats, right? And your body is really good at being able to use these fats for energy. It can make ATP from them pretty easily. All right, and then the last important fat, well, one of the last ones, is a phospholipid. So we mentioned that phospholipids form the cell membrane. They have 2 fatty acid tails, and then they've got this phosphate group head. The phosphate group head is polar, meaning it likes water, and the tails are non-polar, meaning they do not like water. So they tend to arrange themselves in this pattern where we've got the phosphate heads on one side, tails, more tails, and more phosphate heads. So the tails kind of orient towards each other to create a fatty membrane around your cells. You've also got steroids. So steroids we find in different forms, right? Your sex hormones, the major components of your bile, some of your vitamins. But most of those come from a base molecule of cholesterol. So cholesterol looks a little like that. And you notice your sex hormones are very similar in structure because they are derived from cholesterol. All right, and then protein. So proteins are really large macromolecules. They're built of smaller amino acids. Proteins have carbon, hydrogen, oxygen, and nitrogen. So we're adding in another element here. So your lipids and carbs don't have nitrogen. Proteins do. That makes them a little different. They serve a lot of different jobs in the body. Energy isn't one of them, right? So they're not a major source of energy like carbs and fats are, but they do serve a lot of important jobs. So when we're making proteins, we're putting amino acids together. So we can have really small dipeptide here, which is 2 amino acids together. We can have a polypeptide, which is 10 plus amino acids. And eventually we get to a full protein, which is a very highly folded specific structure. And remember, structure equals function. So proteins are one of these examples where the structure has to be just right in order for it to perform its function. So here's an example of protein structuring. We've got to get all the amino acids in the right order. Then those amino acids are going to be attracted to each other in a way that the chain will fold or coil. And then those folds and coils wrap around each other to the point where we've got this very specific 3-dimensional shape. Right? And this is hemoglobin. So that's a type of protein that helps you carry oxygen in your blood. So really, really important that the structure be just right in order for it to do that. Proteins, if they're not folded right, it means they've denatured. So denaturation is when a protein basically melts. Right? The bonds that were holding it in that very specific shape broke, and it's no longer holding its form. So a denatured protein can't be put back together. You just have to dismantle it and start over. One of the major things that proteins do for you, and they create structure in your cells, but they also have really important functions like your enzymes. So enzymes are all a type of protein. So enzymes are proteins. They are a type of protein that can catalyze a chemical reaction, meaning speed up a chemical reaction. You would not be able to maintain your metabolism without all the enzymes you have that are doing their job in your cells all the time. And they're fast too, right? They can do multiple hundreds of reactions per second inside your body to maintain your metabolism. And it's due to the very specific shape of an enzyme. So enzymes have active sites. Active sites are where substrates can fit in. A substrate is just the molecule that the enzyme is working on. So in this case, it's taking these two little molecules and it's putting them together to form one bigger one. So it's helping that chemical reaction go from the reactants here to the product. And then our last major macromolecule is the nucleic acids. So nucleic acids, these are really large organic molecules. They contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. So now we've added in a fifth element here. So that's what makes the nucleic acids special. You really only have a couple different types of nucleic acids, whereas you have thousands of proteins in your body, thousands of enzymes. Nucleic acids, you've got DNA and you've got RNA. And they are each composed of smaller molecules called nucleotides. This is what a nucleotide looks like. It's got a sugar, phosphate group, and a nitrogen-containing base. So DNA is important because that's the recipe book for how to build you, right? A DNA has all the genes in it that code for the proteins and the enzymes that carry out all the work it is to create and maintain a body. RNA is a helper molecule. So it is a shorter lived, sort of transient molecule that helps share the message of DNA to the rest of the cell. Because DNA itself is kind of like sequestered in the nucleus and it's highly protected and doesn't really leave or does never leave the nucleus. RNA can carry that message outside to other parts of the cell. So here's what a DNA looks like. So DNA stands for deoxyribonucleic acid. It is a double helix. It's got these two strands that meet in the middle and are bonded by hydrogen bonds across the middle here. And then those two strands kind of twist around each other. So that's what makes it a helix, right? It's sort of twisted. Okay. This is RNA or ribonucleic acid. Ribonucleic acid is usually single-stranded, so we just have the one strand here. And then the last thing we'll mention in the nucleic acid category here is ATP. So I've mentioned ATP a few times already. It stands for adenosine triphosphate. We're including it here because it's basically just a single nucleotide, but augmented. Right, we've got a 5-carbon sugar, got a nitrogen-containing base, and a phosphate group, but in this case, there are 3 phosphate groups, so triphosphate, so adenosine triphosphate. These extra phosphate groups here are really good at storing energy. So the bonds that hold these together here store a lot of energy, such that when I break off one of those phosphate groups, I release a burst of energy, and that's what ATP is good for. I think I'm going to minimize here. So there's still a lot to cover, but we're definitely not going to get through it in the next few minutes. So let me just kind of tell you the basics. The rest of this unit gets into different tissues. So we mentioned that a tissue is just a group of cells that have a common origin, similar function. So epithelial tissue, connective tissue, muscle tissue, nervous tissue. The rest of the PowerPoint gets into those different tissue types examples of those tissue types, what their functions are. And then it moves into the first one of our body systems, which is the integumentary system. So integument is going to include the skin and appendages like the hair and the nails, so accessory structures. So all of that is covered in the integumentary system unit here. So I highly recommend kind of reading through the rest of the PowerPoint here along with your learning maps as you're studying for the week. Any questions about anything we've covered today or the setup of the course?
So this isn't related to the course, but have you ever heard of Crash Course?
Yes, I have.
Crash Course Anatomy and Physiology is what's gotten me through each of my anatomy and physiology classes.
Yeah, Hank Green is amazing. So those crash courses are really useful. And you'll even see some crash course content embedded in your learning maps. I highly recommend. Sometimes they get into a little bit more detail than we will in this course. But in addition to crash course, another one is Amoeba Sisters. Have you ever heard of that one?
No, but will you post it in discussion for me or in the announcements? Because I know Crash Course helps, and that's actually what I went to get through the chemistry lesson
Perfect. Yeah,
because
so
I haven't seen it yet in the learning content. I was like, okay, where's my Crash Course?
well, I think it shows up eventually, but I'll post some links. Amoeba Sisters are really good. They tend to be a slightly like simpler version, and they're really cute little videos. But I've got some ideas for ones I can post for you. So I'll put links in the discussion of some videos you can use. Also, this video will be uploaded to my YouTube page. So you can, I've got a bunch of pre-recorded lectures there as well. So you can just search Professor Calabrese at Herzing University to find my page, but I'll post links as well.
That works.
Right.
Thank you.
Of course. Any other questions?
No.
Ooh, the door just blew open behind me. It's windy. All right, then I will stop the recording and post this. All right, I hope you have a wonderful day.